So in the first step of the TCA cycle, we have oxaloacetate combining with acetyl-CoA to form citrate. This step is catalyzed by an enzyme known as citrate synthase. This is a Claisen condensation, so H2O is involved, and we will be hydrolyzing the coenzyme A off of the acetyl group. And this step is highly exergonic. It has a very large negative delta G value, so it's going to be a spontaneous reaction. So here we can see the structural form of this step, and we can note that the parts labeled in orange on the acetyl group is what is going to be added to the oxaloacetate to form the citrate. So this depicts the structure of citrate synthase. The first step of the TCA cycle is a regulatory step, and citrate synthase is highly ordered. So that means that the oxaloacetate must bind first to the citrate synthase. Once the oxaloacetate binds to the citrate synthase, the enzyme undergoes a conformational change, and it creates a opening for the acetyl-CoA to bind. And once the acetyl-CoA binds to the citrate synthase, it undergoes a conformational change into a more compact form that is better able to undergo catalysis. So it is highly ordered. First, oxaloacetate must bind, then acetyl-CoA binds, and then it forms a compact structure. So in taking a look at the mechanism, we can note that there are three important amino acids in citrate synthase, and these three amino acid residues work together to allow the acetyl to combine with the oxaloacetate. So we can note that the three residues are histidine 274, his 320, and ASP 375. This ASP 375 has an oxygen with a negative charge. That means that it has three lone pairs. It has an extra lone pair. So that lone pair is going to attack the hydrogen on acetyl-CoA. So this is acetyl-CoA. So when the oxygen attacks that, nucleophilically attacks that hydrogen, the electrons in between the carbon and hydrogen are going to go to the carbon. But carbon needs 4 bonds. So what it's going to do is that it's going to form a bond between its adjacent carbon. So the electrons lost right over here allow for the bond formation with the adjacent carbon and acetyl-CoA. So essentially those electrons are going right there. So now this carbon, it has 5 bonds. So one of these bonds it's going to break, and it's going to be this double bond oxygen. It's going to lose its pi bond character. So 2 electrons are going to be going to the oxygen. So oxygen is going to gain a lone pair and then gain a negative charge. So once it has that lone pair negative charge, it's going to attack the hydrogen on the his 274. So these lone pairs are going to be going back to the nitrogen. So what happened is that we had this ASP 375 that has an oxygen with a negative charge, so it has an extra lone pair that attacks this hydrogen, and the lone pair that is left behind forms a double bond between the adjacent carbon and acetyl-CoA. When that happens, one of the bonds in this carbon breaks, and oxygen gains a lone pair. When it gains a lone pair, it will attack the hydrogen on to his 274, and then the lone pairs from that hydrogen will be given to the nitrogen. So that is the first. That is the first step of this mechanism. What is important to note is that this histidine, both of the histidines exist in their fully protonated form, so they have this positive charge character. So in this step, we can note that the AsP375 has gained the hydrogen that it got from the acetyl-CoA. So this hydrogen was over here, but now it belongs to AsP. So this AsP is now in its resting state, and it is no longer a part of the rest of the mechanism. So we can also note that the double bond has formed between carbon and carbon. Oxygen has gained this hydrogen, which was originally on the histidine 274. So in the next step of this mechanism, the double bond of carbon-carbon will nucleophilically attack this carbon on oxaloacetate. So this causes this pi bond to break, and 2 of the electrons will go to the oxygen. So oxygen will gain a negative charge, and it will have an extra lone pair. That lone pair will attack the hydrogen on histidine 320, and this lone pair over here is going to go to the nitrogen. What is important to note is that when the nitrogen gained this lone pair, it lost that positive charge character and it formed a double bond with its adjacent carbon. The same thing will happen in the case of this histidine. When it loses its hydrogen, it gains a lone pair, it will form a double bond between its adjacent carbon. So while that is happening, this lone pair on the nitrogen will regain its hydrogen. So previously it lost its hydrogen to oxygen, but now it's going to regain its hydrogen through its lone pair. So this lone pair of electrons is going to go back to oxygen. So this double bond will reform because the extra lone pair will drop back down. So to draw out the exact arrows of this mechanism, we can note that this double bond attacks this carbon, which causes oxygen to gain a lone pair. So these electrons are essentially attacking this hydrogen. This lone pair of electrons goes back to the nitrogen, and while that is happening, this nitrogen will regain its hydrogen. So in the next step, we can note that this nitrogen has gained that lone pair, it has formed a double bond between its adjacent carbon, and it has lost that positive charge character. We can also note that this part is the acetyl part, and this is the oxaloacetate part. We have formed a bond between the 2 molecules. As we saw previously, the acetyl attacked this carbon. The double bond has reformed between carbon and oxygen. And this nitrogen has regained its hydrogen. So it lost that double bond and it regained that positive charge character. So in the next step of this mechanism, we will see how that water is involved that we initially saw in the very beginning. So water is added to citrate synthase, and the lone pair on water will attack this carbon on the acetyl part of this citrate-CoA molecule. That will cause one of the lone pairs to go to oxygen. So oxygen will once again gain that negative charge. It will have that extra lone pair. So here we can see that H2O molecule has bound to the carbon. Oxygen has gained a negative charge. This molecule is highly unstable, and this step occurs very quickly. So the double bond character will return because one of the lone pairs will quickly drop back down, reforming that double bond. So carbon can only have 4 bonds, so one of the bonds is going to be lost, and it is going to be the coenzyme A. So this bond will be broken. Sulfur will gain a lone pair, and as sulfur is leaving, it will grab one of the hydrogens from the water with its lone pair that it has gained from right over here. So then we can see that the double bond between oxygen and carbon has reformed, we can note that as the coenzyme A was leaving, it took that hydrogen with it from that H2O. The most important thing to note over here is that H2O had 2 hydrogens. One of the hydrogen was given to coenzyme A. The other hydrogen should be over here, but it is not. That is because in physiological conditions, the pH is 7, and since this acid has a very low pKa, it will exist in its deprotonated form. So we will not draw that hydrogen. So to summarize, we can note that the lone pair on the oxygen on AsP375 attacked the hydrogen on the acetyl group. That caused the oxygen to lose its double bond character and a double bond to form between the 2 carbons. The oxygen then nucleophilically attacked the hydrogen on histidine 274, and the lone pair was between the hydrogen and the nitrogen was given to the nitrogen. The nitrogen will then lose its positive charge character and form a double bond with its adjacent carbon. The double bond on the carbon will then attack the oxaloacetate carbon, and the double bond on oxaloacetate will break, and the oxygen will nucleophilically attack the hydrogen on histidine 320, and the lone pair between the nitrogen and the hydrogen will be given to the nitrogen, and then this histidine will lose its positive character and form a double bond with its adjacent carbon. And then in the last step, we see the H2O coming in and nucleophilically attacking the molecule, forming a temporary highly unstable intermediate. And then as the double bond between the carbon and oxygen is reformed, the coenzyme A is going to leave the molecule. As it is leaving the molecule, it will take one of the hydrogens with it, and then we are left with citrate.